It's my pleasure. My pleasure and honor to be here to share our new study. And during the talk, I hope I can just close the video, then people can focus on the slide. And in the Q&A session, I will turn on my video again. Great. So today I would like to show everyone our new study about how the age tumor microenvironment limit T cell control cancer. And first, I want to start from introduce some important aging in cancer research. So as we have known that aging is associated with the cancer incidence, and in the figures from the Nature Aging 2022, they collect and summarize some data from 2015 to 2019. Then we can find the cancer incidence really increase with the aging, especially when the people age after 55, you can see the rest of from 25 to 75 or even 100 very quick. Second is there were several paper or study already showing that aging can actually return the tumor progressions and the metastasis. And then one of them, the reason could be the change in the tumor driver mutation, or it can be the alteration from the immune or physiology system. And moreover, they also including the variations in the therapeutic efficacy and the side effect. Then, however, we know that the mechanism is still unclear and still have many open questions here. So we face that the impact of aging on cancer can be multifaceted or complex. Therefore, we're thinking maybe we should focus on the tumor microenvironment first to make it more simple. And compared to the young tumor microenvironment and the age tumor microenvironment, we think they will have four difference. Is they may have the cancer cell senescence, genomic alterations, or immune aging, or the stroma or other tissue aging, including cytokine, chemokine, or the metabolite or other factor change. So today, I want to show you the study that we focus on the immune aging, especially CD8 T cells, and we try to show you and how we understand how the HTME limits CD8 T cell control cancer. Before that, there was one question we want to ask: how do we prepare the right aging in cancer model for our study? So there was a review coming up in cancer cell that showing that actually in the talking to about aging cancer model, there were different type, including maybe you want to hematopoiesis, epigenetic, or the more immune aging part. They may be based on your study purpose. Maybe you need to prepare a slightly different model. And our study more focused on immune aging, and several studies already highlight that in fact, when the mice nature aging to 10 to 14 months, although it's not geriatric, so which geriatric means very old, usually means 18 to 24 months, but actually that 10 to 14 months mice already can be considered as an immunological age. So here is some evidence. So actually immune aging can start earlier than when mice was about 10 to 14 months, and it's like the 40 to 60 weeks old. And there was evidence showing in the thymus that you can find in the left part, that you can find the number of the thymic cells will drop down, and after week 40, actually look very similar to week 100. On the right side, there was a human data from how the structure of the cortex inside of the thymus change. You can find the size changing will really limit the maturation of the T cells. So you can find a really sudden drop down when the humans is around 40 to 50, between 40 to 50 years old. And so we're thinking that the immunoagent really start earlier. And in our study, we also take a look on the spleen from different age of the mice, from 5 weeks to 55 weeks. And we use a flow cytometry to checking the PD-1 or TIM3 marker, which more like the, you can say age-associated exhausted or the mature like these T cells. And in here, we can find there was more the PD-1 positive, TIM3 positive increase when the age over the 68 weeks old. And for those naive T cells was dropped down also at the same time. So the both evidence showing that actually the immunological agents start earlier than we expected. And so we started trying to establish the young VSH marine tumor models, and we try to equivalent to the human age. So here is the plot figure from the Jackson library and mapping how in the upper side the mouse age to the lower side of how the human age is. Because the previous evidence was thinking we can look at from the young mice around 10 weeks, 10 to 12 weeks, is sitting in the 20 or 30 years old human. And then we also want to pick out some age between the middle age and old age. And we pick out a 68-week old, and that is mimic the human around 55 years old. And in the previous cancer incidence plot, I also showing when people older than 55 years old, the cancer incidence increase. So our model looks like this. We start from 10 weeks to 68 weeks, and we're giving the BCC over is a kind of the myeloma models, and waiting 15 days and we take down the mice. And the first things we find is really the tumor size increase with the age increase. We can find the 68 weeks old mice have the biggest tumor volume. And this, the bigger tumor size things is also we can find in the different models. Is the LLC, is the Lewis lung carcinoma models. We can find very consistent results. Moreover, maybe some people already find that it just come out in the cells yesterday. There was the paper from the John Cargans and from the first author, Daniel, showing that when you're giving the tumor to the mice, it's the same BCC over. When the mice get older, like 68 weeks and 92 weeks, they have the worst survival and tumor controls, and which is really a consistent result to ours. And then after we know the agent promote tumor growth, so we want to understand how the change in immune cells. So we use the same model, take down day 15 and see the immune cell change. And we take a look for two models, both models, and take looking at the CD8 T cell, CD4 NK DC, and also the macrophage and monocyte. We only find very consistent decrease in both models for those CD8, NK, and DC. So we start interesting to why only those three immune cells are decreased. And actually, they already have several paper or study or mention how important is CD8 CDC1 NK network that was important for T cell priming and differentiation. Then this network actually comes from a very famous cancer immune cycles that was proposed by Harold and Shana in very almost 10 years ago. And more people are looking at this cancer immune cycle and even deeper inside the tumors and see how the CD8 T cells, CDC1 NK cell crosstalk here. And we also have some data but because of time, I cannot show you here, but we really find that the interaction between those three immune cells was decreased in different aspects. So because we know this network is important for T cell priming and differentiation. So the next question is you want to know how the CD8 T cell priming change or the T cell stage during the differentiation. So I will show you the first part first. So before I start, I want to just highlight the speaking to T cell priming expansion. I want to use the cancer immune cycle as an example to show you how it works. So to make the story short, so first is when tumor dying, cancer cell die, that was released on cancer cell antigen and maybe be updating by the DC or the APT antigen presenting cells. Then we'll go to the tumor, the lymph node, and help the CD8 T cells priming activations. And for those, CD8 T cells may able to traffic back to the tumor and recognize the tumor very specifically and kill the tumors. So this comes the cancer immune cycle. And for our study, we really find that we take a look for two different models, persisting ALC overall, and looking at the tetramer-positive CD8 cells. This means the those tetramer-positive CD8 cell can specifically recognize the overall on the tumor. So we find low tetramer-positive CD8 cell was reduced in the tumor in the age cohort, and also those CD8 cells also reduced in the tumor drained lymph node. And because I'm showing you this CDC1 is important for CD8 cell priming, so I will start looking at how the number change when the age of the mice increase. We really can find when the age increase, the CDC1 proportion will drop down in the disease. And also there was reduce of antigen presenting related molecule like the CD40, 86, and class 1, 2 was reduced and in the age CDC1. And in order to further confirm that, we do the single cells. We collect all the CD45 intratumor immune cells and looking at the DC subset, we really can find, compared to the young and age, the age one will have a downregulation of this B2 type 1 for that's very important antigen presenting genes there. So we want to confirm this antigen presenting function of HCDC1 in vivo. The experiment we decide is maybe we can try to mimic mRNA vaccine, or you can say mRNA cancer vaccine. So what we did is that we giving the whole mRNA, the lipid nanoparticle, on day 6 in our model. And when we give that, in the young mice, we really can find they have the better tumor controls. And also they have expansion of CD8 cell in both tumor and tumor drain. And however, for those age mice receive the OVA LNP, they doesn't show the better tumor controls. And also we didn't find the expansion of those CD8 cells in both sides. So we start to make a small conclusion here, say thinking probably the HCDC1, not only the proportion or number drop down, but also those HCDC1 fail to prime expand CD8 cells as young. So here is a small conclusion is the first part, I think, yes, we find impaired CD8 cell priming and expansion. Then we start to move on. The second is how the CD8 cell state change. And before I start here, I want to give a little background introduction about why the CD8 cell absorption. So the T cell differentiation in cancer is very unique. So if you see the right part, cartoon is the usually naive T cells will become affected T cells, will probably clean the firewalls, all those things, then go into the memory. However, they also can become exhaustion if in chronic infection, for example, HCV, or in cancer. So in cancer, usually in the tumors, then the naive T cell being affected level going to more exhaustion CD8 cells, which means still have some function, uh, compared to the affected line of losing their cytokine production or proliferation capacity. Moreover, recent years, we also looking deeper in those exhaustion CD8 cell and define they actually have different stage and subset. So those exhaustion CD8 cell actually start from progenitor and going to the transitory and maybe will go down to terminal exhaustion CD8 cells. And here I usually call progenitor T port and T trend and T turn. And the difference between that is the progenitor cells will have more the self-renew capacity and also have a better tumor controls for the long term. And terminal is more like they're already losing a lot of function, or you can say more similar to dysfunctional CD8 cells. And here is an example about how it looks like in a single cell. In the young mice, usually in a single cell, you can find progenitor, terminal, transitory, and here is a naive cells, and here is a dividing CD8 cells. However, in the age mice, we find there was a drop down of this progenitor, terminal cells in the age tumor, but increase this very unique T-TAT cells, because over here in the plot. And this T-TAT is the called tumor infiltrating age-associated dysfunctional CD8 cells, so we call T-TAT. So it's a very unique and new subset in the age tumor. And we want to understand where it comes from and how it comes from. So we decided two experiments. One experiment is maybe these T-TAT cells come from because the aging in the T cell itself. So we decide the experiment for co-transfer young and age OT1 to the mice and young mice and see whether we can find these T-TAT cells. Another is why if the actually the environment cause those T-TAT cells. So we transfer the young mice, young OT1 to the young and age mice and see how the environment change and see how the cell state change too. On the first part, we also do single cells from young to young or the age OT1 to young. We don't really find the generation of the T-TAT cells, but we find the reduction with the progenitor. It looked like those age OT1 more easy to go into the terminal and lost the progenitor phenotype. So we think the intracellular change in age T cells is not driving the T-TAT cell stage. And second is why if we transfer young to the young and age mice, then we can find when the young OT1 transfer the age mice, we really can find this new T-TAT subset showing there. And we also find the reduction of the T-terminal, which is similar to what we found in the endogenous models. So we think it's actually the HTME promote this differentiation to the T-TAT cell stage. And we're wondering how different between these new subset compared to the other subset we already know, for example, the progenitor or terminal. So we have the same experiment, day 15, we're sorting all the subset and do ATACs to look at how the epigenetic regulation change. So this very surprise to us is even though the difference between the young and age progenitor or young and age terminal, but they all showing in the same area and grouping together. However, the t-tet are really different from each other. And second is we do some functional test. We sorting out tetramer positive T cell from young age mice and transfer to another young mice for another 8 days to see how the in vivo controls. So we can find these T-test cells is not showing the beta in vivo tumor control like the progenitors T cells. And another assay we did is we also sorting out those subset and then in vitro culture with the B16 oval tumor. We also can find these T cells not like the terminal solid T cells can have better or remain the cytotoxic ability. So can we find those T cells in human? That's the next question I want to ask. Then we actually collecting the data from different published data set and merge it together become a human pan cancer analysis. And you can see here is we can find more like dividing cells or the interferon gamma memory light or T-tet, and here is the terminal. And with the age increase, we actually can find there was increase of this T-tet subset in the human too. And when we mapping the mouse T-tet gene signature to the human one, we can actually find the very high the correlations, which means they just double confirm that the subset what we find in mice is similar to what we find in human. So the second part of conclusion is that yes, we find some dysfunction CD T cell stage is called T-tet. And because the impaired priming and those dysfunctional CD T cell stage, so final we want to ask is there any way we can improve the CD T cell control of a tumor? So one thing come to our mind is whether CD40 agonist is a good option to reinvigorate those HCDC1. The reason is because previous publication from Roberts, there's already showing how the agonist CD40 antibody to activate DC to help the CD T cell control tumors. And also, not only his team, there were several papers I just put to highlight to here, also showing that the CD40 activates DC can really increase some critical T cells like stimulation cytokine like IL-12 and was important for CAT cell activations. So then we start to using CD40 agonist antibody. Our experiment design is giving on 2 doses on day 2 and day 7, and also analyze on day 15. In the young mice, we can find better tumor controls. And surprisingly, we also can find the better tumor control in age mice. And for those age mice inside the tumor, for those CDC1, we also can find the increase for the CD40, 86, and MESCAS1-2 expression. And most important is that we really can find the expansion of CD T cell now in both both tumor and tumor-genetic. And then we start next to look at how the differentiation look like after treatment. We find after the CD4 treatment, we can find they will reduce progenitor, terminal, exogenous CD cells, including the T-tet dysfunctional CD cells, but increase more effector-like CD cell after treatment in both young and age mice. And I want to highlight another study here before I go into summary is the actually from their teams also find the DC activator can improve T cell mediated tumor control in age mice. And the figure that's shown here is before they test that, they actually check the PD1 or PD1 with the CTLA4 in young and age mice. So you can see here, 8 weeks compared to 20 weeks doesn't have difference. However, when the age increase, becomes 68 weeks compared to 8 weeks, you can really find the age mice is not really benefit for the tumor control or the survival. For those 2 as old young mice, 8 weeks receiving PD1 or the combination therapy, they can. And so what I did is that I really think that this is the major driver to maintain the T cell mediated tumor control. So what they did is giving the age mice for this the whole tumor lysis with the LPS, kind of the total reserve for active stimulator and PGPC. They can find for those age mice receiving the active DC hyperactivator can really control the tumor better. So a little similar what we found in giving the CD40 agonist. So here I want to give you the summary. The first part is we find the ATME really can limit the NK cells with CC1 and CD8 T cells crosstalk and impair their primary expansions. And second, we find this very unique age-associated dysfunction stage, here we call TTED, that was different from the resolution we know in the young mice. And we also find the myeloid target therapy for the CD40 agonist can reinvigorate the CDC1 and improve T cell priming. And the fourth is I don't have time here, but I want to highlight here what we found and what we will do in the future is we actually find some T cell intrinsic defect, and those including those age T cell actually have lower proportion cytotoxicity ability or the DNA damage resistance. So I want to end here, and thanks for everyone who working in this project, and special thanks for my mentor Deb and Nir for their support, and thank you for you.
Thank you, Alex, for this beautiful presentation. So if anyone has any questions, please raise your hand or type it in the chat, and I will read it for you. In the meantime, Alex, I was looking at your data and you show very clearly that it's something in microenvironment that shapes T cell responses. However, there's a population there from the T cell progenitor that is not rescued when you transfer age T cells to a young mice. Can you comment a little bit on that result you have?
Maybe I should share my screen,
Yeah,
share
sure.
my slide again,
Sure.
and you can
when you do the transfer T cell model from the age to young, there's a
yes.
the TJ progenitor, and you map the results.
You mean this one?
Yeah,
yes.
the T progenitor that seems that it's not rescued when you transfer to.
Oh, you mean the reduction of the?
Yeah.
Yes.
Can you comment a little about what could be happening there?
Yes. So, as I mentioned in the final summary, we actually checking some cell intrinsic change. We find the age OT1 compared to young, they are more easy to go into the terminal and have DNA damage inside the cell already. So that reason we think when those age OT1 transfer to a tumor, they were easy to lose the progenitor or more easy to go into the terminal compared to the young one. For the single cell, for the crown, only showing the progenitor reduced, but we also have some flow data showing that.
And
yeah.
Thank you. And just one more question. So did you check? I mean, you showed that there's a deficit in antigen presentation by CDC1s, but I was wondering if it's also deficits on migration from the tissue to the draining lymph nodes. Perhaps also they are not not only presented antigen well, but also they're not migrating. Do you have any results regarding that?
Yes, I guess you mentioned about the T's, this one, right? You mentioned about migration here. Yes, this is a really good point. So actually, we have preliminary results. So we using different models is called IL-12 reporter mice. Then they can able to catch the mature DC. Then we really found those kind of mature DC not only reduce in the tumor, but also tumor draining flow. For those mature DC, basically they carry the antigen from the tumor to tumor draining flow. Yeah, so you are correct. Yes, they also reduced.
Okay, thank you. So I have one question from Anna and then Dr. Aditya. Anna, go ahead.
Thank you very much. It was an excellent talk, an excellent work as well. I have a question whether you also could follow up for, I don't know, development of metastasis or something, because I found it very interesting that you see those differences in the age of adult mice. But I also thought that, you know, we are always told from a human perspective that the older we get, the most likelihood we have to get cancer. And I wonder if you could check other aspects like metastasis and so on. Thank you.
Yes, that's a really good point. So yes, we're looking forward to check that. And because the model we use for B16 and AOC, it's not easy to find the metastasis, but we will use other model for KP or use another way to inoculate tumors. Is a very good point. Thank you.
Dr. Aditya, if we want to open your mic.
It was a very great presentation, sir. And I have one small basic doubt. When we have this CD20 agonism, will you okay with combine with the CD20 agonism with PD-1 inhibitors? Like in India, we are having this cancer therapy trials. So what is your expert opinion about it?
Yeah, this is a great point. You mentioned CD40 with the checkpoint inhibitor, right? We thinking to doing that in the future, and we hope to give you guys update soon. Yeah.
So in our government hospital in Chennai, right now we are planning for it, and we have incorporated it, and so we will give you the video also, sir. It's a pilot project which we are doing it. Thank you,
That's
sir.
great.
I don't know if anyone in the audience has more questions. In the meantime, Alex, can I share my presentation? I just want to invite the audience to our next Open Box Science seminars. We have really, in the upcoming weeks, we have two super seminars. The first one is a START protocol to the use of these cave mice to photo label and track in vivo. So it's going to be super seminar as well. And additionally, we have a neuroimmunology seminar regarding TRIM2 and Alzheimer's diseases from the Weizmann Institute of Science. Follow our social networks and you'll find our Zoom registration link as well. So if there's no more questions, thank you very much. Thank you, Alex, and good luck in all your endeavors.
Thank you. Thank you, everyone.
All right.
All right.